Gram staining was first introduced by a Danish bacteriologist named Hans Christian Gram in the year 1882. At present time, Gram staining is one of the most crucial staining techniques in microbiology because not only it is used to identify bacteria, it is also used to diagnose infectious diseases caused by bacteria. In identifying bacteria in Gram staining, different stains or reagents are used to distinguish two kinds of bacteria. Gram-positive bacteria, which retains the purple stain color, and Gram-negative bacteria, which exhibits pink or red color. For the slide preparation, a permanent marker was used to draw a circle at the center of the glass slides. The circle was drawn on one side only of the glass. This served as the marker for the smeared sample. After that, the glass slides were labeled using paper tape and a pen. For the collection of human flora, sterile swabs were used to get saliva and vaginal discharge. The samples were smeared in a circular motion as thin as possible with at least a 1.5 cm distance in the top and bottom area. At least 2 to 3 samples were prepared, and the slides were labeled using a paper tape. For the collection of colonies from a solid culture, 1 drop of distilled water was placed in the glass slide. Inoculating loop was sterilized with the help of a flame from the alcohol lamp. After a few seconds, the inoculating loop was used to inoculate a single colony from the solid culture media. The sample was spread by smearing in a circular motion on the glass slide, and the smear was made as thin as possible. The smear was air dried, and the labels were checked. The same procedure was done for the remaining samples, and each time, the inoculating loop was sterilized and was cooled inside the biosafety cabinet. Before the staining process, each slide was heat fixed using an alcohol lamp and cooled down. In Gram staining, different stains or reagents are used, such as crystal violet, Gram's iodine, decolorizer, and safranin. A sufficient amount of crystal violet was dropped in each smeared sample in the slides, ensuring that all samples are covered for 1 minute, and then they were washed with water. A sufficient amount of Gram's iodine was dropped into each slide. After 1 minute, they were washed away with water. The slides were decolorized until all stains were washed away. Then, they were washed again with water. After safranin was dropped in each slide, they were soaked for 1 minute before washing them with water. A A light compound microscope was used to examine each stained slide. Each slide was observed under all the objectives including scanner, low power objective, high power objective, and oil immersion. After performing a proper sequence of gram staining procedures with cultured and human body samples, both gram-positive and gram-negative bacteria were observed through microscopy of the stained samples. For the culture media, both specimens A and C, which were from the CS Dean's Doorknob and CS Biology Laboratory Teacher's Table, contain gram-positive bacteria, while specimen B from library's female CR contain gram-negative. Specimen A has cocci, specimen B has both bacilli and staphylococci, and specimen C has staphylobacilli. For the human flora specimens, vaginal discharge contains gram-positive bacilli, and both saliva and vaginal discharge contain gram-positive cocci. In terms of color, no specific observation was made except for their translucent or whitish appearance. Though gram-positive bacteria have a peptidoglycan outer layer, this thick layer easily absorbs antibiotics and disinfectants, thus making them easier to kill. On the other hand, though gram-negative bacteria have a thin peptidoglycan layer, this layer does not absorb the materials around it, thus protecting the bacteria from specific attacks. Moreover, though both typically survive for a long time, gram-positive bacteria can survive longer on dry surfaces, while gram-negative bacteria tend to survive longer on wet surfaces. This could be one of the factors why gram-positive bacteria were found in the Dean's Doorknob and the Teacher's Table, while gram-negative bacteria were found in the library's female CR, specifically on the bowel of one of the cubicles. With this, it could be implied that the bacteria found in the CR are more dangerous than the bacteria found in the other two locations. Thus, preventive measures such as hand washing before and after use should be taken upon going to such a place.